Hello, botanists. Welcome back. So our focus today is going to be on the process of secondary growth. Now, we've already looked at primary growth in both roots and stems. And remember, in primary growth, that's how new sections of the root are going to get produced. That's how lateral roots or branch roots are going to get produced. That's how root hairs are going to get produced. Thinking about the stem, primary growth is how new sections of the stem are going to be produced, how leaves are going to be produced, how branches are going to be produced. Well, what is secondary growth? Well, secondary growth is going to be how our stems widen. Now, roots can go through this process too. Our focus is more going to be on the stem. This is going to be responsible for root or stem widening, how they get thicker. Did I spell that right? It looks really funny. Widening. I have too many there. There, widening. Widening of our root or stem system. And ultimately, this is going to be how wood and bark are going to be formed. And I think you know about those things, probably not from a botanical perspective, but just from being a human and being out there in the world. Well, what we're going to do is we're going to make some drawings to model how this process is happening. And where I want to start is I want to start by making a diagram of a eudicot stem. And this is going to look very similar to what we saw in lab a couple weeks ago. And this is just going to be a eudicot stem cross-section. And the reason I'm making this is to just review primary structure, what it looks like once primary growth has occurred, and then also to show where our secondary meristems are going to originate. Because overall, it's the activity of those secondary meristems that's going to produce secondary growth or widening in the stem system. Okay, so let's draw just a little circle here. This is going to be that stem cross section. And remember that outermost layer is going to be the epidermis. Our vascular tissue is going to be in these regular bundles that are in a ring. And so I'm just going to draw two, three, four. Now, since this is a eudicot, these are going to be in a ring. They're not going to be scattered throughout our ground tissue. The inside of each one of these bundles is going to be primary xylem. So I'm just going to shade that there in red. The outside of each one of these bundles is going to be primary phloem. So I'm going to shade each one of those in blue. So these are just those bundles of vascular tissue. Primary xylem on the inside of the bundle, primary phloem on the outside of the bundle. And I'm just going to abbreviate xylem with X and P, just because we've seen those terms a bunch now. I don't think it's going to add too much to write them out each time. All right, so that's our vascular tissue, right, in these discrete bundles. Well, last but not least, we've got our ground tissue. Remember, if we're talking about this ground tissue that's out here, in between our bundles and the epidermis, that's the cortex. If we're dead center in our stem, inside of those bundles, we're looking at the pith. And then finally, if we're in between those bundles, right, in these regions right here, that's the region of ground tissue that we just call pith rays, because they're rays of pith that radiate in between our bundles of vascular tissue. Now, this is all review. This is all stuff that we've seen before, right? We've seen models like in these diagrams. We've seen the real thing in lab a couple weeks ago. What we're going to do now is to show physically in space where our secondary meristems are going to originate in this cross-section. Now I'm going to skip this to side and talk about those two secondary meristems really quick, give you their names, and then I'm going to add them to my drawing. So we've got two secondary meristems. And overall, it's the activities of these that are going to accomplish secondary growth. Their function is to really produce secondary tissues. Now, you've seen primary growth already. Remember, in primary growth, we have our primary meristems, our protoderm, procambium, and ground meristem. Those produce primary tissues. You've already seen that happen. While in secondary growth, we're going to see secondary meristems producing secondary tissues. And there's going to be two secondary meristems. We're going to derive them one at a time. The first one that we're going to have originate is going to be what we call the vascular cambium. Now, the vascular cambium, we're going to draw it originating in just a second. The vascular cambium is going to form from residual procambium that's left inside those bundles of vascular tissue. So it forms from that residual procambium within the bundles. It also comes from cells within the pith rays. So cells inside those pith rays are going to dedifferentiate, give up their function, and become meristematic and link up with that residual procambium to form this complete ring, the vascular cambium. And overall, once that's done, we're going to have this ring, right? This ring of meristematic cells all the way around. So you've got this ring of meristematic cells, right? Remember when you see that word meristem, you should think lots of mitosis, lots of cell division. We've got this ring of meristematic cells all the way around that's going to divide to the inside and to the outside of that ring. And so when that ring of meristematic cells divides to the inside, it's going to produce secondary xylem. All right, so that ring divides to the inside, secondary xylem gets produced. And when it divides to the outside, secondary phloem is going to get produced. And that's going to happen first. Well, let's draw this physically in space on the model that we just drew, right, this one. All right, here's our bundles of vascular tissue. That hard black line that I have separating my primary xylem and my primary phloem, that's that residual procambium. All right, so that right there, that's that residual procambium, just meaning that it never grew up, it never turned in to either primary xylem or primary phloem. Now that's already sitting there waiting, that's already meristematic. But then cells in these pith ray regions right in here are going to dedifferentiate, give up being pith ray cells, probably doing something like storage, and become meristematic. Parenchyma cells can do that. That's one reason why they're so cool from a developmental perspective. They can give up their function and take on a different one. In this case, becoming meristematic. So cells inside those pith rays are going to dedifferentiate, give up their function, and link up with that residual procambium until we've got this ring of meristematic cells all the way around, right? All the way around the circumference of our stem here. And once that's happened, right, then we've got our vascular cambium. Just this ring of meristematic cells all the way around the circumference of our stem here. Right, that's going to happen first, right? It's going to get active and start producing secondary xylem to the inside and secondary phloem to the outside. Well, our other secondary meristem is something called the cork cambium. And we'll add it to our list in just a second, but I want to show physically in space where it's going to originate. That cork cambium is going to originate from the outermost layer of cortex. So right there's our epidermis. If we just move in one cell layer, that's where our cork cambium is going to come from. So that's going to form from the outermost layer of cortex. I'm just going to draw a little circle all the way around, getting as close to the epidermis as I can. That's that core cambium. That's where those cells are going to dedifferentiate, become meristematic, and form this meristematic ring, just like the cells in the pith rays did. All right, well, let me add the core cambium to our list of secondary meristems here. So our secondary meristem is going to be the core cambium. And as we've just seen, right, it initially forms from the outermost layer of cortex. And it, like our vascular cambium, is going to be a ring of meristematic cells. And just like our vascular cambium, it's going to be dividing to the inside and the outside. And based upon whether those cells are produced to the inside or outside is going to affect their development, affect their fate, right? What they turn into. When the ring of meristematic cells divides to the inside, that's going to produce something called phylloderm. Now, phylloderm isn't something that we've seen before, not like we've seen xylem and phloem, right? Secondary xylem and secondary phloem are going to function just like primary xylem and primary phloem. They're going to do exactly what you'd expect. Right? Secondary xylem transporting water, secondary phloem transporting sugars. Well, phylloderm isn't something that we've seen before. How it's going to function is it's going to function kind of like a secondary cortex. And a lot of what the cortex does in the stem is it serves as storage. That's how the phylloderm is going to function in a stem that's going through secondary growth or widening. So phylloderm is going to get produced when the core cambium divides to the inside. When the core cambium divides to the outside, cork is going to produce. Now, you have a concept of cork, right? Just from cork boards, corks in things like wine bottles. I've seen cork shoes. I have some in my Birkenstocks. There's cork blocks in yoga, right? Cork floors. That's true botanical cork. Cork is going to function kind of like a secondary epidermis. Now, the epidermis, remember, is the outside usually. But what happens in secondary growth is that we get all this growth and expansion from the interior of the stem, and that's going to stretch apart the cells of the epidermis and kill them. Same thing with our cortex. We're getting all this growth and expansion from the inside, and that's going to stress out those outer layers, and so it's going to get replaced, right? The cortex gets replaced by the phylloderm. The epidermis really gets replaced by the cork. And so the cork is going to be responsible for preventing too much water loss and also invasion by insects and things like that. All right, well, let's see what happens when a little bit of secondary growth is occurring. So I'm going to make another stem cross section. Now this is going to be a woody stem with just a little bit of secondary growth. With I abbreviate just like that. And so we're seeing some secondary growth, not a crazy amount. And where I want to start this diagram or model is I want to start it from the vascular cambium. All right, there's our vascular cambium. This ring of cells that's meristematic, that's dividing to the inside and the outside. When it divides to the inside, secondary xylem is going to get produced. And it's going to be produced in these layers. I'm just going to add a layer here. All of this, and I'm going to shade it in. All of this is my secondary xylem. So that's going to get produced when that vascular cambium divides to the inside. Remember, when our vascular cambium divides to the outside of that ring, secondary phloem is going to get produced. So I'm going to add a layer of that. It's going to be that secondary phloem. Right on the outside of that vascular cambium. Well, remember that vascular cambium formed inside those pith rays and literally inside our bundles, literally in between our primary xylem and primary phloem. Once that vascular cambium gets active, that's going to separate our primary xylem and our primary phloem. And our primary xylem is still going to be in these little clumps next to the pith. I think I drew 4 in my last drawing. I'll try and do 4 in this one too. If these are just those little clumps of primary xylem, our primary phloem is going to be just opposite of those. Because remember, they were next to each other. They've just gotten separated because that vascular cambium has added secondary xylem and secondary phloem. That primary phloem is literally just getting pushed towards the outside by the addition of those secondary vascular tissues. All right, well, what about outside of that? Well, if we go way out, if it's a little bit of secondary growth, we're probably still going to see some little bits of epidermis. Now notice I drew that as broken, right? At this point, it's probably broken and dead. And that's just because we've got all this growth and expansion from the inside up. And forming the new outside are going to be the cells derived from that cork cambium. So I'm going to still use that purple to make it parallel with my last drawing. Right, our epidermis is dead. It's in the process of being shed. It'll eventually flake off the tree. And I'm going to use this new term periderm. Think about this term. Peri means around, just like it did in pericycle. Derm means skin, right? Just like it does in epidermis. The periderm is just the collective term for the cork cambium, and phelloderm. So it's all of those collectively. Sometimes it's hard to tell them apart, and it's useful to use this collective term. We're going to use it because a lot of the slides that we have in our collection, we won't be able to tell between those easily, and so we'll use that collective term. And so that new periderm is forming essentially the new outside. Well, the vascular cambium is going to be more active than our cork cambium in here in this periderm. And what that means is that we're going to get the addition of lots more secondary tissues, lots more secondary xylem and secondary phloem than that cork cambium can keep up with. And what that's going to eventually mean is that our first periderm, that first one to originate, is going to get stretched apart and break just in the same way that our epidermis is. So what we're going to start to see is we're eventually going to see new periderms form. And each one of those just represents a new paradigm. A new one that's forming inside of the old one. Just because the old one is getting stretched apart, it's breaking, it's dying, just like the epidermis did before. And you've seen this before. You've seen how some trees have kind of flaky bark that you can kind of peel away in these layers. What you're probably peeling off is you're peeling off those paraderm layers. All right, so these new paraderm layers are going to form in this kind of patchwork fashion. They're going to form from inner layers in this patchwork fashion to replace paraderms outside of them that have already died. All right. This will be the end of part 1. I'll make a part 2 in just a minute.